All right, so new topic, conjugation, dienes, and resonance. All of this is going to introduce you to a new and famous reaction called the Diels-Alder reaction, which the author of the text absolutely loves because it's a famous reaction used in all kinds of syntheses of complicated molecules, and we will learn that. But some of what we talk about today is also going to be helpful in understanding how you see. So just as a introduction, we've talked in the first part of the class about making pi bonds and combining two p orbitals together to get two new pi molecular orbitals. And the lowest energy orbital is the one that we get from adding the two p orbitals together side by side. That's the plus combination. If you actually go to Spartan and draw this and look, this is what that orbital looks like. So when you see a diagram like this, you have to remember that what we're really talking about is this. You can tell that this is a bonding orbital because there is electron density in between the two nuclei. You can also recognize it as a bonding orbital because we get it from the plus combination of these p orbitals. We get the minus combination up here, and there is a node in between the two. We concluded before that the pi orbital is the HOMO, whereas the pi star is the LUMO. Looking vaguely familiar? Questions about that? Anything? Okay, well, if you calculate the difference in energy between HOMO and LUMO, and this is a number from Spartan, 7.5 electron volts. You don't need to write that down or remember it, just putting that in context. If you take that energy and convert it into wavelength, the kind of photon that would promote an electron from HOMO to LUMO has a wavelength of about 165 nanometers, which, if we go to our friendly electromagnetic spectrum, the 100 nanometers is sort of in the ultraviolet. Okay, so molecules that just have one double bond typically aren't colorful. They don't absorb in the visible range of the spectrum. Now, we should talk about what happens when you start to line up more than just two p orbitals side by side. And I think we talked briefly early on in the class about the allyl anion. And we talked about how if we call these carbons 1, 2, and 3, how resonance tells us that we've got double bond character between 1 and 2 and between 2 and 3, and that we have negative charge on 1 and 3, but never 2, right? Because of these two conclusions, we concluded that each of the atoms in this anion are sp2 hybridized, and therefore that each has an unhybridized p orbital above the page and below the page that are lined up with each other, parallel to each other, where they can interact. Is that sounding vaguely familiar? Okay. And we talked about how you can take these three 2p atomic orbitals and mix them together to get three new pi molecular orbitals. And what these looked like, and these are the kinds of things that I'm not going to ask you to be able to draw from memory, but I do expect you to be able to look at them, look at these orbitals and find the nodes and figure out their relative energy levels. The three orbitals we get from this picture look like this. The lowest energy one we get from adding the three p orbitals together, all the plus combination. The middle one is kind of weird. It has a node that coincides with carbon atom 2. That's required actually by the math of MO theory, and so I won't require you to generate this from scratch. But in this orbital, the orbital density is on carbons 1 and 3, but never on 2. These aren't two separate p orbitals. It's one orbital, and an electron in this orbital would be delocalized between carbons 1 and 3. Then the highest energy one's the one where they are all sort of out of phase with each other. And so to order them in energy, we count the nodes. We've got 0 nodes here, we've got 1 node here, we've got 2 nodes there. So they are in order of energy. And then we fill up these orbitals with the available number of electrons. These are the electrons involved in pi bonds or adjacent lone pairs. And we would see, okay, we've got 4 electrons involved in resonance. And so we fill the 2 lowest energy orbitals, and then we can conclude HOMO and LUMO. And we saw that the picture that this MO theory gives us coincides exactly with what resonance tells us. The lowest energy electrons account for the double bond character between 1 and 2 and between 2 and 3. Right? Those electrons are shared all across. Whereas the HOMO here accounts for the lone pair that's on carbon 1 and on carbon 3, but never on carbon 2. Okay, I think we did that very briefly, so that's just the quick summary. We can continue to do this with additional p orbitals, and so the next iteration would be, what if we have 4 carbons that are all conjugated to each other? And that occurs here in 1,3-butadiene. You have carbons 1 and 2 that are involved in a pi bond, but you have 3 and 4 that are also involved in a pi bond, which means on each of these atoms you have a p orbital that's orthogonal to the sigma bonds coming out of the page towards you with the blue colored lobe, and going back into the page away from you with the red colored lobe. The red versus blue represent wave function sign. And this is a situation when you have p orbitals on adjacent atoms. Another way of saying that is a contiguous array of p orbitals on sp2 hybridized atoms. You have a situation where conjugation can occur, and that means we're going to mix these 4 p orbitals together to get 4 new pi molecular orbitals. Now at this stage, I've copied in the orbitals from Spartan for 1,3-butadiene. And again, this is not the thing I would expect you to draw from memory. However, if I did show you orbital drawings schematically and I hadn't already ordered them in energy, I would expect you to be able to tell me which orbital was lowest in energy, which was highest in energy, and so on. And I would expect you to be able to tell me which was the HOMO and which was the LUMO. I'm just drawing them schematically here to the side so you can see how I would represent them in a drawing, say, on an exam. I'm pointing out that in the lowest energy orbital, we are adding all of the p orbitals together in phase. That's the plus combination. There are no vertical nodes. In the second combination, we're adding these 2 together in plus combination, these 2 together in minus combination. These 2 together also plus relative to each other, but the sign switches in the middle. And then in the third one, you have 2 nodes, one here between 1 and 2, and this one here between 3 and 4. And then the highest energy one, they're all out of phase. So what this tells you, and we can count the number of electrons that are involved in pi bonds in 1,3-butadiene, this tells you that the lowest energy electrons are spread out over all atoms 1, 2, 3, and 4. and the next highest are also spread out over atoms 1, 2, 3, and 4, but the wave function goes to 0 in between 2 and 3. We would also say that the LUMO is here, it's the pi 3 orbital, whereas the HOMO is here, it's the pi 2 orbital. Okay, any sort of questions about that? So if I show you pi orbitals for a more complicated system, remember, you're just going to count the nodes, arrange them in increasing order of nodes, and determine how many pi electrons, and then tell me what HOMO and LUMO is. Yeah?
Why is the LUMO the pi 3 bar, not the pi 4?
Why is the LUMO the pi 3, not the pi 4? The pi 4 has 3 nodes, the pi 3 has 2 nodes, and we simply run out of pi electrons. In 1,3-butadiene, we've got 2, 4. We fill the 2 lowest energy ones, and then we're done. Now, stay tuned because orbital shape is going to matter here, but I want to point something out about vision, and that is that the difference in energy between the HOMO and the LUMO here is now a smaller amount. It was 7.5 electron volts before. Now it's 5.5. And that moves the area of the spectrum where this molecule would absorb a little bit to the red direction. Not hugely. We're still in only the 200 nanometers. We still absorb it only in the UV light. But as we get up to 400 nanometers and so on, we're going to start to be in the region of visible light. And the trend is that the more conjugation you have, the more p orbitals we link together side by side, the smaller the gap between HOMO and LUMO can be. And eventually you get into the region where photons in the visible area of the spectrum promote an electron from HOMO to LUMO. So I just want to show you an example of a molecule that does that. Again, this is not anything to scramble and try to copy down. Again, we are in such a crunch for content, but this is so important, like you see all the time, right? And so you should understand how it happens. This molecule is called 11-cis retinal. And it's got, the numbering here is not the numbering of the carbons whereby you get 11 cis. Here's the cis double bond, but I'm just illustrating that you have now 12 atoms involved in conjugation. That's 12 p orbitals, okay? And that's going to narrow the gap between HOMO and LUMO to a distance in energy that corresponds to photons in the visible region. This terpenoid, don't worry about, you may learn about terpenes in 352. So this molecule is found, is conjugated to a protein called rhodopsin, which is essential for vision. So here is a brief summary of vision from, looks like Pearson Education. I would have thought I had lifted it from Wikipedia. In any case, here's your eye. This is not an anatomy class, so I don't know. You guys know more about this than me. Here's the retina, okay? And within the retina, you've got different layers of cells. You have the retinal pigment epithelial cells. These are about loading and charging the rod and cone cells with the appropriate photoreceptor discs. And then the rod and cone cells signal via synapses to various cells called these things, which eventually goes to the optic nerve. Okay, that's all the biology I know. But inside the rod and cone cells, you have these photoreceptor discs or lamellar particles. And inside those particles, you have membranes with individual opsin or rhodopsin proteins. Okay, so we're just increasing the levels of magnification, right? We went from eye to retina to a rod/cone cell to the photoreceptor discs and into the membrane of those discs. And this is a membrane that's just folded back on itself. But embedded in that membrane is the protein that detects light. And here in a little red squiggle represents that 11 cis retinal. You'll learn about proteins later. This is a 7-helix transmembrane protein that spans the lipid bilayer. It has an extracellular domain and an intracellular domain. But the key part is this retinal molecule that's embedded inside the helices that span the membrane. And the retinal is loaded onto the protein via chemistry you'll learn about in 352. It's an imine bond. We take a nitrogen from the protein and we replace the oxygen in the carbonyl. Looks weird now. You'll figure it out in 352. But basically, we have this system of 12 p orbitals and therefore 12 pi molecular orbitals. And here I'm showing you the LUMO of 11 cis retinal. You can see that it spans most of those atoms. Okay? And if you measure the gap between HOMO and LUMO, it's around 3 electron volts, which, going back to our spectrum, starts to absorb in the purple region of the spectrum, about 425. Now that's just 11 cis retinal on its own. Molecules like this, when they get loaded inside the protein, the environment of the protein affects the HOMO and LUMO gap, and that changes slightly the area of the spectrum that you can absorb in. And so this is why you have, I forget which ones see color, but you have some, help me out here, somebody that knows this. Which is it, rod or cones that see color? Cones? Okay, good. Well, now I know, and I'll forget. But this is why you have cone cells that can sense in the red and in the green and in the blue, and then your brain integrates those 3 colors to see all of the colors that we see. Well, what happens to have this vision signal occur? You have this gap between HOMO and LUMO. So you get UV light, or you get rather visible light that promotes an electron from HOMO to LUMO. That temporarily weakens some of the pi bonds and weakens the conjugation, and that allows for what's called photoisomerization. And this is where the less stable cis double bond snaps back to the more stable trans double bond. Remember, I told you that rotating around double bonds would require you to break a pi bond? Well, we've already sort of done that by promoting an electron from HOMO to LUMO. And that change in shape, notice that as we zoom in on that protein, you can see the curvature in shape. Imagine the molecule is anchored to the protein here. If we isomerize to trans there, that's going to swing all of this up. And that movement creates a change in shape of the protein, which is coupled to some downstream signaling that results in vision. All right? And all of that was happening all of the time since you first emerged into the world and squinted into the light and started crying, right? And as we've been talking about this, your eyes have been absorbing billions and billions of photons, and this has been going on. Incidentally, after you isomerize to trans, that molecule of rhodopsin is no good anymore. And so what happens is discs that have already absorbed light get sloughed off into the retinal pigment epithelial cells where the membranes, where the rhodopsin proteins are reloaded with a new cis retinal molecule and then traffic back to the rod and cone cells for another round of vision. And you'd think that that would take some amount of time, and yet we don't usually generally get the spinning dial of doom in our eyes saying, hold on, loading, right? Pretty cool how all of that works. So now at parties, you can tell friends that, hey, did you know that the reason we can see those flashing lights is because of absorbing of a photon, promoting an electron from HOMO to LUMO, followed by a photoisomerization from cis to trans? And at that point, they will wander away from you. But if somebody's interested, then you'll know you've got a fellow nerd, right? And then you can, the second question you ask them is, do you know how soap works? And if they know that, they'll be even more likely to be OChem friendly. Okay, so that's the vision story. And I think that's cooler than the Marvel vision story. Anyway. So now we're going to talk about a particular reaction involving dienes, which are and in particular 1,3-dienes. That is dienes that involve conjugated double bonds. And this allows me to also tell one of my favorite OChem jokes. Don't look at that yet. Oh, dang it, I spoiled it.
Too late.
Too late. All right. Help, I'm dying. All right. Later on in 352, I won't get a chance to do this with you guys, so I'm going to do it now. In 352, you'll learn about a concept called aromaticity, and it's about having a conjugated ring with the right number of electrons. And one of my only talents in life is drawing a 7-membered ring, which I've been told looks like a sad robot. So, I don't know, I think he kind of looks happy, but maybe a little bit better. Okay, enough jokes. We have no time for jokes. There's always time for jokes. The reaction we're going to introduce is called the Diels-Alder reaction. And it was discovered in 1928 by German chemists Otto Diels and Kurt Alder. And they discovered the following reaction. They started with cyclopentadiene, and they added a molecule that they came to call a dienophile, meaning the thing that reacts with the diene. The key structural features of the dienophile are the presence of a carbon-carbon double bond, olefin or alkene, we can call it, and then also the presence of these ketone electron withdrawing groups. And we'll talk about why those are electron withdrawing a little bit. So you've got the diene and the olefin, and you put them together, and you usually need heat. Now, the organic chemist will usually use the symbol delta to mean heat. I don't know why, other than it resembles a fire. So, ooh. And the product they get, you're going to love this, is a bicyclic compound. That looks like that. And yeah. All right. Now, is that not something out of your nightmares at this point? Yeah. You don't have nightmares about the demigorgons from Stranger Things anymore. The 4 mouth flap thing just doesn't bother you. You're totally desensitized to that kind of horror. But behold! All right, so let me show you where the atoms end up. The 5 carbons in the diene, which I'm going to number 1, 2, 3, 4, and 5, end up here. 1, 2, 3, 4, and 5. The atoms in the olefin, which I'm going to just call, we'll label purple. We'll start 1 and 2 with the 2 carbons of one of those 2 olefins, 1, 2, 3, 4, 5, and 6. Those end up here, 1, 2, 3, 4, 5, and 6. Okay, so what has changed? Anytime you learn a new reaction, it's useful to focus on what has changed. we lose the pi bond between 1 and 2 in the starting material. We lose the pi bond between 3 and 4 in the starting material. And we lose the pi bond on the olefin between 1 and 2. But we make a new pi bond between orange 2 and 3. And then crucially, we make new sigma bonds between orange 1 and between purple 1. And of course, sigma bonds are more stable than pi bonds. And then we make a new sigma bond between orange 4 and purple 2. All right? So these are the bonds that we make. I'm sort of highlighting them in red. All right. So that was the initial discovery. Go ahead. There's no pi bond between 4 and 5. Rather, it's a sigma bond. And that is still here. Carbon 5 is, yeah. Dienophile. Oh, no, we didn't. I am so sorry about that. So I'll pause for you to correct your notes. Dang it. Do you see this pi bond here between 5 and 4? I forgot to put it there, but it still stays there. All right. So a more simplified version of this reaction would be to take a diene like this and a dienophile like this. And again, I'm going to number 1, 2, 3, 4 in orange, and then 1 and 2 in purple. And in this reaction where we add heat, we get the following product. And I want you to compare this simple example with what I showed you previously. Here's a new sigma bond, there's a new sigma bond, there's a new pi bond. 1, 2, 3, and 4 in orange. 1 and 2 in purple. Okay, does that maybe clarify just a little bit what went on above? Okay, so as you get older and further on into organic chemistry, recognizing structural features in products of reactions becomes really useful, because then on synthesis problems you sort of know, oh, I want to end up with this product. I think I'm going to need to use, say, a Diels-Alder reaction. And the key structural feature of the Diels-Alder reaction is that the product is a cyclohexene. Do you see 1, 2, 3, 4, 5, 6 atoms in this ring? And do you see that there is a double bond, and only one double bond in the ring? That is the key structural feature. And you can find it up here as well. 1, 2, 3, 4, 5, 6. 6-membered ring containing the double bond. Everything else is just details, and I'm going to show you how to deal with those details. But this is the core of the Diels-Alder reaction. And because it forms a 6-membered ring, you can tell why organic chemists are obsessed with it, because we love 6-membered rings. They're fun to draw, and they also don't have a lot of ring strain, and so they're very reasonable, straightforward to make, and they show up in all kinds of natural products. Okay. Question so far about what happens? Yeah?
What is the purpose of this reaction?
What is the purpose of this reaction?
Like learning about why we need to remove alcohols, how to handle alkenes.
Right.
What is this chapter?
What the heck is this about? Well, we're going to talk about how this reaction requires conjugation to work. but the reaction is also synthetically useful. I don't necessarily, well, why not? I mean, in 1952, several years after Diels and Alder discovered their reaction, and just a couple years after they won the Nobel Prize for it, Professor R.B. Woodward at Harvard, he's sort of the godfather of modern synthetic organic chemistry, did a similar Diels-Alder reaction. He used the following, and you can sort of see the structural similarities with what he did to what Diels and Alder did. This was his dienophile, and here were the two atoms in that dienophile. And then he did a reaction with this diene, and here are the atoms there, and he was able to make the following molecule, and you're going to see the six-membered ring cyclohexene product. And oh, whoops, that's not that's just a methyl group. And then through several additional steps, converted this product into a steroid called cortisone. And so that was, like, I think the first, and of course there's many more complicated steps going on here, but this was, I think, the first organic synthesis of cortisone. And the idea that we can take important biological molecules and make them whenever we want and change their structures however we want is an important factor in modern drug discovery. So, where did this key six-membered ring that he made end up? Here is the Diels-Alder ring here. And I'm going to number in orange where the original atoms of the diene and the dienophile were. And then that ends up in the product here. All right, so anyway, it's synthetically useful is basically the answer. All right, now all of these bonds, all of this bond changing happens at the same time. So this reaction is concerted. Its energy diagram would involve a single transition state. And you expect this reaction to be thermodynamically favorable because sigma bonds are more stable than pi bonds. Here's something that's useful to think about, just as an aside. I told you the reaction's exothermic, meaning you release heat because the bonds you form are more stable than the bonds you break. Why do I have to add heat to do the reaction then? Go ahead.
To break the bonds because it takes heat.
Takes heat to start breaking bonds. Yeah, effectively you're adding the heat to get over the energy barrier. Okay, remember, getting over the transition state doesn't have a lot to do with the difference in energy between starting materials and products. So even though this reaction is exothermic, we're providing heat to get it to go, not because the reaction absorbs heat. It doesn't. It releases heat. But you need to put in some to get over the transition state and get to the product. All right. So because all of these things happen in the same concerted step, we can draw the mechanism like this. Pi bond moves to form a sigma bond. This pi bond moves over to form the new pi bond between 2 and 3. Then this pi bond moves over to become the new bond between 4 and 2. Alternatively, you can draw it the other way. You can have the arrows go around clockwise versus counterclockwise. Nobody cares. Nobody will ever say boo, depending on which way you draw the mechanism. It's okay. It's a concerted mechanism, and notice there's 3 pairs of electrons involved in this, 3 things going on at once. So I want to show you the HOMO and LUMO orbital picture so that you can get a sense, or see at least, how this reaction, like the ones you've learned before, involves the HOMO of the nucleophile attacking the LUMO of the electrophile. Okay? So let's just do this simple example, and we're going to just remind ourselves briefly of the butadiene, or the diene molecular, pi molecular orbitals, and then we're going to remind ourselves briefly of the molecular orbitals of our alkene or olefin. So the diene, we have pi 1, pi 2, pi 3, and pi 4. If we're, and remember that we drew the pi 1 orbitals with all the shading in the same direction. The pi 2 has one node between atoms 2 and 3. The pi 3 has a node between 1 and 2, and then another node between 3 and 4. And then pi 4 has the maximum number of nodes possible. All right, and we, in our discussion previously, said that pi 2 is the HOMO. And for our discussion today, we're going to think about the diene as the nucleophile. And what I'm going to tell you is only the story of 2 of the 3 electrons involved. And there will be alternate ways of thinking about this reaction, but this is one that's pretty useful. Okay, for the olefin, we have the pi and the pi star. The pi looks like 2 p orbitals adjacent to each other. The pi star has a node in between atoms 1 and 2 of the olefin. We okay? Just a quick summary. All right. So let's see whether it's reasonable for the HOMO of the nucleophile, the diene, to attack the LUMO of the electrophile, or the olefin. So instead of drawing this diene flat in the page, I'm going to draw it standing on edge as though we're looking at it from its side. So here's 1, 2, 3, and 4. All right? And then we'll draw the olefin also standing on its side. At the same time, I'm going to try to draw the hydrogens that are on the diene. And then here's atoms 1 and 2 of the olefin. And again, it's standing on its edge. So we're drawing the hydrogen atoms there. Okay? That's our perspective, is we're viewing these things from the side. Let's draw the HOMO of the nucleophile. And this is going to involve, again, the pi 2 orbital, which, if we draw it, is going to, let's use, I don't know, blue and red, is going to have a node in between carbons 2 and 3. So that node would bisect the butadiene if we were looking at it from the top, but now we're looking at it on its edge. I realize that's messy, but do you get the gist? Okay. Now let's draw the LUMO on the olefin. And notice that we can draw that LUMO such that the portion of the LUMO that is on carbon 1 lines up with, in phase with, the portion of the LUMO that is on orange 1. In other words, we can have productive bond-forming overlap between the pi 2 orbital on the nucleophile and the pi star on the olefin. Do you see how they have the same phase, same sign? Okay. Similarly, on carbon 4, orange 4, I can line up with the pi star on purple carbon 2, again with the same same sign, a bond-forming productive overlap. Okay? So it turns out that for the diene and the olefin, the shape of the orbitals is compatible with the reaction. Some people like to think about this in terms of symmetry. Briefly, you could see that the pi1 has mirror symmetry, whereas the pi2 has what's called C2 symmetry. C2 is 180 degree steering wheel action. Grab either end of the molecule and then spin it 180 degrees and you get the same thing. So you can see that if we spin this orbital 180 degrees, you would get exactly the same thing. Similarly, the pi star of the LUMO also has C2 symmetry, whereas the pi has mirror symmetry. So the LUMO and the HOMO have the same type of symmetry, which guarantees that their end carbons will overlap with each other in phase. Okay. Now that's just the story of these two electrons from the HOMO sort of dumping themselves into and attacking the LUMO. You can also, if you want, go through and show the HOMO of the olefin attacking the LUMO of the dienophile, and you will notice that they also have the same symmetry. You've got a match between the symmetry of the HOMO of the olefin and the symmetry of the LUMO of the diene. Okay, so there's multiple things going on here. So it turns out that two of these electrons become one of the sigma bonds. The other pair of electrons becomes the other sigma bond. And then the lowest energy pair of electrons just stays and becomes the pi bond. Do you see how that pi bond between 2 and 3 is sort of embedded in that orbital? Okay. So orbital symmetry is a big deal. There's a whole field of OChem that has to do with what kinds of reactions are allowed versus not allowed. For fun, and I know none of you actually want to have fun at this point in the course, but you should, for fun, you could convince yourself why this is not allowed by orbital symmetry. See whether you can do the same kind of thing with the HOMO of one olefin and the LUMO, the pi star of the other, and see whether there's any way to get them to come together side by side with matching orbital phases on each one, and it doesn't work. So it turns out you can't do this reaction just by adding heat. And there's a longer story there, but one that we don't actually need to talk about, other than to say that this reaction does happen all the time, but it requires UV light. And that type of reaction is one of the things that happens to you when you get sunburned. So someday in biochem, I think you're going to learn about DNA damage and thymine dimers. And it involves a reaction like this that's not allowed by heat, but is allowed by UV light. All right. So with that in mind, you've got the picture of how the bond forming happens and how it's concerted. Now we want to talk about the basic issues associated with this reaction. Okay, so 4 issues that we need to master. One is enantioselectivity. And I will just tell you there is no enantioselectivity in this Diels-Alder reaction. We're going to need to talk about something called diastereospecificity. That's going to relate to the structures, how the structures on the diene and the olefin influence the substituents on the diene or the olefin influence the substituents on the product and their stereochemistry. We're going to talk about another issue called diastereoselectivity, and this has to do with something called endo versus exo. And then finally, we will talk about regioselectivity. So, enantioselectivity, can you get one enantiomer over the other? And the answer is no. So here is a slightly more complicated situation. We have a diene, here is our diene, and here is our dienophile or our olefin. And we want to know, when we do the Diels-Alder reaction, can we get one enantiomer over the other? Here is the 6-membered ring product, and of course I could have the methyl group here, or I could have the methyl group pointing down away from us. All right? And the answer is no, you can't. You get one of these products so if we put this diene on its edge and have the methyl group on, I guess we're calling it at this point, orange carbon 4, then that methyl group sort of pointing out of the page towards you, and you've got hydrogens going in that direction. And if we have the olefin approach the diene from the right-hand side, let me put in dotted lines the bonds that we're going to form. Do you see how when we approach from this side, this hydrogen on carbon 4 is above the ring that we just formed, whereas the methyl group is below the ring we just formed? You see that? So that's this product when we approach from the right side. If in contrast we approached the diene, if the olefin approached the diene from the left-hand side, sorry, some of those protons are sloppy. Here is our new bonds that we formed, and again, it looks like the methyl group is below the new ring that we formed, and it is, but now the double bond is there, and when we do the friendly neighborhood pancake flip, we can see that we get this product. Okay, so there's no enantioselectivity because the faces of these planar systems are exactly the same. You approach from the left versus the right, you get both enantiomers. Okay. So if your product is chiral in a Diels-Alder reaction, you have to get both enantiomers. Now, any of you who are interested in working in the Michaelis lab, they do an enantioselective version of the Diels-Alder reaction, but they have to use a catalyst that is chiral. Okay, second issue, diastereospecificity. What do we mean by that? So far, the olefin and the dienophile that the olefin and the diene that we have used have been fairly simple, not a lot of substituents. And the question here is, what if we start to put non-hydrogen things on one or both of the partners? So CO2ET stands for an ester group with an ethyl group attached to the oxygen. And in our olefin, our dienophile, carbons 1 and 2 of the olefin both have these ester groups on it, and the relationship between those two groups is cis. Because this reaction happens in a concerted fashion, there is not time for rotation around around this carbon-carbon bond between purple 1 and 2. And so when we form the new ring, those two groups have to be on the same side. Okay? So the product that we draw must show those two ester groups on the same side. Now, do we only get the up product? No, actually, it's totally fine for us to draw the olefin this way. The reason I didn't do that, though, is because that is actually the same meso product. But the diastereospecific part is the substituents on the diene are cis, the substituents on the olefin are cis before. They must still be cis relative to the new six-membered ring that we formed after. Okay? That is a key feature. Naturally, if instead you had used the trans-alkene, you would expect to see those ester groups trans to each other in the six-membered ring product. And if your product is chiral, remember, you have to get the other enantiomer too, which I'm indicating with that plus minus symbol. Okay? That's the diastereoselectivity part that's easiest to see. The second part is a little bit more difficult to see, though it's easiest to see if we go back to look at our original DieneAlder reaction. By the way, notice that the substituents on carbons 1 and 2 here are cis to each other on this ring, right? You can tell that because both the protons are up, which means these groups are down. Now, let's look at the diene. There are two positions in the diene. You have this sort of inside positions, and then you have these outside positions. Now, in a normal diene that's not cyclic, this would have been a proton here and a proton there. But you see in the product that, again, relative to the six-membered ring that we formed, the two things, or in this case, the one thing that was inside is above the ring, and the things that were outside are below the ring. Now, the above and below isn't the important part. The important part is the two outside substituents have to be cis to each other, and if you have two inside substituents, they have to be cis to each other. And I will show that to you in more detail next time. We only have a little bit more to do with the DieneAlder reaction, and then we're going to move on to radical chemistry.